Hi, good morning, everyone. Thanks to the organizers for inviting me for a short talk today. It's a very great pleasure for me to be here, well, even if I'm not here physically. Um, so my name is Raphael. I'm a postdoctoral researcher associate at the Oak Ridge National Lab in the US. And a large part of my research I'm going to present today was actually performed at the University of Pretoria in South Africa. In the Forest Molecular Genetics group led by Zander and co-supervised by Stephen Hussey. So I am a molecular biologist with initial training in molecular biology, biotechnologies, bioinformatics, and for a few years I had a growing interest in computational approaches that make use of artificial intelligence. And I must warn you, I'm not an expert in that field at all, but I had a chance to test some of these methods and try myself to some of these methods, and I got very excited. So today my presentation won't be that much about showing the results, but rather introducing some of these technologies to you, and some of you might be aware of this already, and hoping it can lead to some interesting discussions and ideas. So I'll be very quick on the introduction, but just to give you some background on why we ended up looking at AI approaches. So as much as most of you are aware already, wood formation is a complex process that involves thousands of genes that have to be expressed at the right time in the right tissues. And to orchestrate the expression of all these genes, the plant uses a specific family of proteins that we call the transcription factors. And these transcription factors are proteins that are able to regulate the transcription of sets of genes that we call the target genes, generally by binding to the DNA into the promoter region of these genes, and then regulating their transcription. So if we look at what we know of these regulatory interactions, and taking the example of secondary cell wall formation, for example, which is a critical step during wood formation, some of the target genes that are directly involved into the cell wall formation were already identified in tree species, largely based on knowledge accumulated in model plants like Arabidopsis. So we kind of know which genes are at the bottom, the ones involved in building the cell wall, but for the regulatory pathways that are upstream of these genes, the transcription factors that are upstream of these genes, we still don't know if the regulatory networks are conserved between a model plant like Arabidopsis that doesn't actually form wood and tree species like poplar and eucalyptus. So looking at Arabidopsis and two tree species like poplar and eucalyptus, for example, we can find some examples of key transcription factors that seem to have similar functions in Arabidopsis and trees. So, but these are only for very few examples. And so it looks like some of the nodes in the regulatory networks might be conserved between Arabidopsis and trees. But we still don't know if the wiring of the networks is conserved. Actually, there is growing number of evidence suggesting that the wiring is not that conserved. It's quite different between Arabidopsis and trees, with probably even plenty of new key regulators that don't even exist in some herbaceous plants like Arabidopsis. And so if we don't know the regulatory pathways that well, that's a serious problem that we are facing, because obviously then we can't anticipate the effect of genetic perturbations on the phenotype. And when I say genetic perturbations, that can be either polymorphisms that segregate into a population, or mutations that we just induce by genetic